Hello, everyone, and good afternoon. Great to see as many participants participating in this webinar, a very interesting webinar with a title of the causes of sperm DNA fragmentation and ways to reduce it. Let me introduce the one who's going to speak on this, Dave. Dave is a director of clinical support, CooperSurgical. He's been clinical embryologist since 1986, managing IVF lab across the UK and consulting internationally. Former chair of the UK Association of Clinical Embryologists and Association of Biomedical Andrologists. He has contributed to key regulatory groups and now serves as a CooperSurgical Director of Clinical Support, focusing on the process optimization, lab design, and training. Well, Dave, thank you for give us a very interesting topic, and the floor is yours. Thank you.
Thank you, Bador, and thank you to all the Middle East team to set this up. I'll just share my screen. Hopefully you can see that. Good evening and good afternoon, everyone. I can't necessarily see you, but hopefully you can see my screen. We're going to talk about sperm DNA fragmentation and what we do in the lab to limit its impact on treatment. What do we, first of all, let's see what we mean about sperm DNA fragmentation, because we need to understand what we measure, what it means, and what the potential impacts are. And we could start right at the beginning with the fact that the sperm is a unique cell in the male body, designed principally to deliver DNA from the male partner to the female partner with the intention to elicit fertilization and embryo development and ultimately a healthy pregnancy. And I'm sure you're all aware that sperm undergo a radical restructuring from when they are a haploid spermatid, when the cell is completely remodeled to form the mature spermatozoan that we're typically used to seeing down the microscope. And of course, one of the key elements here is that the genetic material, the chromatin, is repackaged, and we know that a large proportion of the chromatin is repackaged so that it's in this donut loop model and packaged really densely with protamines, which replace the histones that we see in somatic cell nuclei. So that transition between histones and protamines allows this dense packaging, but that, whilst protective, doesn't rule out some potential for DNA fragmentation, which we'll come on to. Now, the reason we see DNA fragmentation, it can come from a number of different sources. It can be due to testicular factors where there's incomplete or defective sperm maturation. And there can be errors in apoptosis, so abortive apoptosis during spermatogenesis. The apoptosis would normally ensure that defective germ cells are not able to go through spermiogenesis and form fully mature sperm. But errors in that can occur. They can occur in the testes or the genital tract, that allow breaks in DNA to be passed through to the mature sperm. And then we have clinical risks and environmental risks that affect the sperm after production. We'll come on to those in just a moment. As I'm sure you're all aware, one of the key factors that we need to keep in mind when we're talking about sperm DNA fragmentation is the exposure to oxidative stress. So this is a mechanism whereby the DNA in the sperm nucleus can be impacted by oxygen-free radicals, reactive oxygen species that cause breaks in the genetic material. Another thing that we need to be particularly aware of is the fact that we talk about sperm DNA fragmentation as if it is a single entity, and it may not be. And the tests that we use, we will often talk about DNA fragmentation testing, and that really is an overarching name for a range of different methods. They don't all test the same thing. Each test is not necessarily, and we have a panel of tests, including the COMET assays, the sperm chromatin structure assay, the tunnel, the terminal deoxynucleotidal transferase nick end labeling assay, and we have the halo test or chromatin dispersion test. And you may or may not be using one of these. They have different approaches. Now, as I've said, I'm sorry this is probably looks quite small on your screens, but it's just highlighting the point that there's a whole range of studies. You can trawl the literature. This is taken from Ashok Agarwal's paper in 2020. But what it illustrates is that you can trawl the literature, and there is a whole body of papers that demonstrate some association between sperm DNA fragmentation and reproductive outcomes. And as you can see, what we find is that people use different assays, the tunnel, the comet, the halo, so on. And they'll look at different impacts on pregnancy outcome, whether it's in IVF cycles or ICSI cycles, whether it's donor oocytes, whether it's swim up and so on. And as you can see in this column, there's a whole range of different cutoffs that have been reported in the literature for different tests, different cutoffs or thresholds at which point the sperm DNA fragmentation test is showing some association with a reproductive outcome being impacted. So that obviously the higher the DNA fragmentation, the more likely it is to impact a reproductive process. But as I've said, different tests will measure different things. This is a really, really nice review by Riba Mainu and colleagues from 2019. So it's not particularly new, but it's a lovely review of sperm DNA fragmentation. And they give a much more detailed and eloquent description of the methodologies and the tests involved than I can give in this talk in the time available. But the key message is that if you do a tunnel, the SCSA, the halo test or comet, they don't detect exactly the same type of DNA fragmentation. And more importantly, perhaps, the different types of DNA fragmentation seemingly have some different effects on the reproductive processes. So if we look at these, tunnel will measure single strand breaks and extensive double strand breaks, and comet, depending on whether it's alkaline, neutral or two-tailed, will measure single strand breaks and some double strand breaks and so on. And there's different levels of sensitivity and specificity in these tests. And some will detect breaks in these MARs, these matrix attachment regions. Now, a matrix attachment region in the sperm nucleus is where the DNA has not been complexed with protamines, and it remains complexed with histones. So these are like the nucleosomes in somatic nuclei. They are complexed with histones, and that's potentially important because we know some of those regions that remain complexed with histones rather than protamines code for genes that are linked to embryo development. So that's an important thing to remember. So we've got these potential breaks in our DNA. They can be exacerbated, compounded, made worse by what we do as men. So we know, for example, that sperm production and the levels of DNA fragmentation is sensitive to what we ingest, whether that's the types of food, whether it's alcohol, whether it's prescribed drugs or recreational drugs even, and linked to that, weight, obesity and body mass index all have an impact on sperm DNA fragmentation. So what we do as men, what we eat, what we ingest is potentially important. Similarly, we know the environment has an effect. So we know there are environmental pollutants which can damage sperm, and that might be related to the job that you do. But more generally, pollution as a whole potentially has an impact on DNA fragmentation rates. We can offset that a little bit by taking oral antioxidants. So again, that sort of relates to diet as well, that if you have a good diet, well balanced, that can be protective, or we can take oral antioxidants, and there's some evidence that that can help offset the worst effects of sperm DNA fragmentation. Then there's temperature. We all know that the testis is at lower temperatures than the body, and that higher temperatures that we see with, for example, taxi drivers, professional cyclists, where there's evidence that scrotal temperature is elevated for considerable periods of time, we know that that has an impact on sperm quality generally and has an impact on DNA fragmentation. Then, of course, smoking. We know how bad smoking is for all sorts of reasons. And of course, it has implications and impacts on the quality of the sperm that the man produces. And of course, that can be seen not only for conception, but miscarriage, and maybe even going through to the offspring as well. And then there's age. We know that the older a man is, the more likely he's going to have elevated levels of sperm DNA fragmentation. As a man just about into his 7th decade, I just know lots of meat doesn't work as well as it did earlier, and there's no reason to assume that that doesn't impact on my reproductive potential as well, of course. So what can we do? What can we do to offset those risks and think about how we deal with DNA fragmentation in the laboratory during medically assisted reproduction? What can we do? Well, the first thing we can do is we can work to isolate populations of sperm with low levels of DNA fragmentation. I've used this slide before, you may have seen it. There are some interventions that we can do in the laboratory and before the sample gets to us that will help mitigate sperm DNA fragmentation. So the obvious one is abstinence. We know that as the period of abstinence increases, the likelihood is that sperm that have been produced for a while and sitting around in the epididymis and the reproductive tract, they can undergo spontaneous DNA fragmentation. So if it's sitting around for a longer time, it makes sense that the levels of DNA fragmentation would increase over time. So the flip side of that, of course, is true, that if we limit the period of abstinence, we can offset that. So if a man ejaculates more frequently, and then we get a sample for use in treatment, we have to be wary that the sperm numbers might be reduced because you've cleared the stored samples in the epididymis. So you might have lower numbers of sperm, but you should have higher quality sperm. And there's some really nice papers show that. There's a particularly nice one from Jaime Gonzalez from the University of Madrid, where he actually reduced the period of abstinence right down to 3 hours. So he produced, the men produced 2 samples on the same day and used the second one. That's probably extreme and risks low numbers of sperm if you want to do conventional IVF, for example. But it illustrated the point really clearly that abstinence is linked to the level of DNA fragmentation. So now we tend not to say 3 to 5 days abstinence, we might say 1 or 2 days, 1 day maybe being the optimal. We need to think about other things that impact DNA fragmentation when we're handling the sample. So I'll talk about sperm preparation in a moment, but we need to think about how quickly we prepare the sample after production, how we store it after preparation, and then we'll talk in a moment about the selection of individual sperm for ICSI. I'm not going to talk about surgical sperm recovery. There are some papers that suggest collecting sperm at the site of production using a testicular biopsy gets sperm with much better DNA integrity, particularly in men with persistent elevated levels of DNA fragmentation. But that data's under question now, and it's a big question mark whether that's a realistic or appropriate way to treat men, I think. So let's talk about sperm preparation and what we do and why we do it. And we'll talk about how that impacts DNA fragmentation, and from there, how that impacts the outcomes of treatment. You'll know as well as I do that the principal aim of sperm preparation is to get rid of all the stuff you don't want and to keep a selected population of sperm with normal morphology, minimal DNA damage, and intact cell membranes, which means they'll function as sperm should. So that means getting rid of the seminal fluid. We know that has decapacitation factors and so on. We want to get rid of the non-sperm cells because we know there's a source of reactive oxygen species that can damage sperm, and the immotile sperm similarly can generate some reactive oxygen species, but they're just not needed anyway. So what we're seeking to do is get this nice selected population of sperm, and then we can capacitate those by incubating them at 37 degrees, and then use them in treatment, whether that's ICSI, conventional IVF, or IUI. And of course, the other thing that is a byproduct of sperm preparation is we don't want to introduce infections, and we want to avoid uterine spasm, which would be caused by, for example, the prostaglandins in seminal fluid. So that's why we have to get rid of that. And that's species specific. Some animals actually the uterus is exposed to seminal plasma. In the human, clearly it isn't. The stopgap is at the cervix. So let's think about the sperm preparation techniques that we typically use. I'm sorry, I'm going to go over old ground. You will know a lot of this, but I'm going to go through it. The swim up selects sperm by their ability to swim out of seminal plasma into an upper layer of medium. That means you generally need good quality samples with decent motility. But we know it reduces the carryover of reactive oxygen species, and we know it samples after preparation that have lower DNA fragmentation. The only caveat being that you have to do it from whole semen. You don't wash first because that's detrimental. Similarly, density gradients, same end goal, that you end up with lower ROS and lower levels of DNA fragmentation. But of course, you're spinning sperm down through a density gradient, so you're selecting based on density of the sperm. And as the sperm mature and they lose their cytoplasm, the cytoplasmic droplets, and the sperm head is becoming more and more dense, then obviously the overall density of those sperm increases. Now we can play around with the density gradients a little bit with how fast we spin it and for how long. But it's worth mentioning again that sperm reach their isopycnic points. Isopycnic point is the level of buoyant density. That's if you spin them down, they'll reach a point where they are at the same level of density as the medium around them. That's the buoyant density point or isopycnic point. And they'll reach that typically if you've got the right setup of your gradients at 300G after 20 minutes. So spinning harder for spinning longer probably isn't going to make a significant difference. And if you want to increase yield, it's better to do more tubes, not spin them harder and longer. Now, we will always hear the story, but centrifugation is detrimental to sperm. And it may be. But it's worth going back to the primary literature and working out at what point centrifugation actually becomes a real risk. So how detrimental is it actually? Well, it can be detrimental and it can cause reactive oxygen species production. But if you go to the primary papers, Mackler and Jacobi, back in 1981, only demonstrated a decline in motility if you go above 580G. When they span sperm at 330G, there was no impact on motility. We also know from the paper by Zinni and colleagues back in 2000, they attributed better DNA integrity after swim up to the centrifugation step that they used in density gradients, but they were using 600G. So this high level. Now, if we look at a more recent paper by Molly Quinn and colleagues in the US, they were using a Zymot device, the Zymot ICSI, and they compared it with density gradient. They found no difference in outcomes, and they were spinning much more gently. So if you spin gently, there doesn't seem to be an impact. So this 300G or thereabouts is probably fine, and you don't see any clear detrimental impact on the sperm. There is a paper again from way back in 1992 by PICSI and colleagues where they did multiple centrifugation steps at 400G and showed that you might have an impact on the acrosome, but they couldn't show an impact on fertilization or cleavage. So we can spin sperm. We just have to moderate the speeds to around, certainly below 500G, 300, 200, 300G. Now there's another way to do it. I'm going to use the Zymot as an example. They're often referred to as microfluidic devices. There are a couple of other options on the market now, but the Zymot is the established one and the one for which we have most data. So these are often referred to as microfluidic devices. We can argue, if you want, about whether it is or it isn't, but the point is these devices rely on a selection of the sperm by their ability to swim through the pores in a membrane between the seminal fluid and an overlying medium. And what we know from the available data is these seem to be easier to do than a standard swim up because the interface is so clean. And you end up with better sperm quality post-prep, and that may or may not yield better outcomes, and we'll come on to that in a moment. So with these devices, you don't need to spin at all if you're doing ICSI. If you're doing a conventional IVF, there's a general recommendation that you wash after prep, and we know they reduce reactive oxygen species. Now, there are other sperm preparation techniques that I haven't got time to go into today. These migration sedimentation chambers, the selection using the charge, the small negative charge on the outside of mature sperm, and there's MACS as well, the magnetic activated cell sorting systems. They all reduce DNA fragmentation after preparation. Does it matter? This is a paper from Rad and colleagues. I could have picked any number. This is comparing sperm DNA fragmentation rates after preparation by standard swim up, pelleting swim up, so that's wash then swim up, which we don't generally advise, density gradient and density gradient versus swim up. And you can see the swim ups do really quite nicely. The density gradients also reduce DNA fragmentation. And it's quite a step down from the raw semen. It's not so obvious in that chart, but we can see it in other papers. This these really show why these microfluidic chambers, the Zymot, are seemingly a step up in their ability to reduce DNA fragmentation. This is a paper from a US group, from Giampiero Palermo's lab, from Perella and colleagues in 2019, where they compared density gradient with the microfluidics. It was a Zymot multi-850 in this paper. And what they showed really nicely, if you just look, you can look at the other parameters, but the one I want to focus on is the DNA fragmentation. So 20% in the raw semen, 12% after density gradient, so reduced, 1.8% in the microfluidic group. So a real stepwise decrease in DNA damage if you select by motility using a Zymot device. And that's really mirrored by this paper from IVF Brussels, where under Nilke de Monk's supervision, this paper is primarily authored by Matezel from last year. And again, they've compared using a Zymot multi-850 against MAX, which we know should reduce DNA damage, density gradient, and a simple wash, simple sperm wash, centrifugation wash. And again, if you look at DFI, the DNA fragmentation index, these levels are lower than you would normally expect to see. 6% in the sperm wash, 2.7% after density gradient, 2.1% after max, and 0.2% with the Zymot device. So again, a stepwise decrease in the sperm DNA fragmentation after this microfluidic sperm separation device. In this case, again, the Zymot multi-850. Now, I've said sperm handling and preparation is important. I'm not going to go into this in any detail, apart from to say that if you should shorten your sample production to preparation time, and you should shorten your preparation to use time as much as possible. And if you can't, you keep them at room temperature, because we know DNA fragmentation increases spontaneously and increases spontaneously more quickly the higher temperature that you get. And this paper, for example, from Rouget and colleagues, these are in men with known elevated DNA fragmentation in a tunnel assay, and these are known men with known low levels. And you can still see after preparation there's a reduction and then a steady increase over time. So use your samples as quickly as you can. Does it impact outcomes? Well, you've probably already been made aware of the Boomsma systematic review. This compared principally swim up and density gradient in IUI cycles and showed no difference. There's a paucity of really strong data. That was the outcome of this particular review. And it doesn't give data on live births, only clinical pregnancy rate per couple. But what it suggests is that once you've got good quality sperm in your post-prep sample, it makes no difference to IUI samples. And similarly, this paper a couple of years ago from Rao and colleagues looked at cumulative live birth rates after IVF and ICSI, comparing density gradient and swim up, and showed no outcome, no differences in outcome, which is perhaps what we'd expect. This is the same paper I've mentioned previously from Molly Quinn and colleagues in the US, comparing microfluidic processing, that's a Zymot ICSI device in this case, compared to a density gradient. And again, they didn't really see any difference in outcomes. But note, that's a Zymot ICSI device that uses microchannels and is designed specifically for very poor samples. It's not the same device as the porous membrane you see in the Zymot multi. You might also, when you look through the literature, find this systematic review of microfluidic devices, the Zymot principally, against standard methods by Ferreira, Alderaldo and colleagues from 2023. And what these this systematic review suggested was that there was no significant improvements in pregnancy, miscarriage, fertilization, and so on. But there's an important thing to say about this systematic review, and that is the standard methods bundle together swim up, density gradient, and density gradient followed by swim up. That's the control group. And in the test group, the sperm separation devices, the microfluidic devices, it includes the ICSI device and the multi devices, so the microchannels and the porous membranes, and they're very different. So I just flagged that up as a word of caution, and that systematic review compares two groups of different approaches to sperm preparation. It may not be forensic enough to actually detail specific differences between methods. The other thing to say, of course, is this is a paper from Denis Sakas's group that highlighted that when they looked at the results, they couldn't find an impact on embryological outcomes, aneuploidy rates, and so on. They couldn't link that to sperm factors. And this is an important point because what we know from all of these data is there is a primacy of the oocyte. Taking into account all of the other things, if you've got good quality oocytes, they will repair some DNA damage from the sperm. If you've got poor quality oocytes, they're unable to do it. So if you look more generally at unselected populations where the oocyte quality might be good, then small levels of DNA fragmentation may not be important, and the oocyte quality is always going to take precedence. Where do we go now? Well, the first thing to say is if you, again, look through the literature, there is a whole body of evidence, and I've picked out a number of studies in this slide, and I'll summarize it quite simply as there are bodies of evidence that show that sperm DNA fragmentation is linked to the likelihood of euploidy or aneuploidy, whichever way you flip it, in the resulting blastocysts, and you can see that in PGT cycles in all of these papers. Now, what we also know from our work that we've done with the Zymot devices, and this is a nice paper from Maria Banti and colleagues last year, where they couldn't show a difference in outcome in terms of fertilization, but blastocyst formation rate, utilization rates, and euploidy rates were better when they used what they called a microfluidic sperm sorting chip. It was the Fertile Plus, that's exactly the same as the Zymot Multi 850, and it showed improved outcomes. Now, when you look more generally at the data, what we find is if you look at the rate of euploidy in blastocysts by the sperm preparation method, you can pick out a whole number of peer-reviewed papers, and what you'll see is that where the asterisks are, these are the ones that showed a statistically significant improvement in the rate of euploidy in blastocysts in PGTA cycles, where a Zymot was used as opposed to the density gradient group. And you can see of the studies here, most of them show an improvement. It's not universal. The Perella, the Leisinger and the Godiwala paper couldn't demonstrate a statistical difference, but the trend is certainly there. And it links to those earlier papers I showed you where the rate of euploidy was linked to sperm DNA fragmentation. And as I've shown you, the rate of DNA fragmentation after a Zymot prep shows a stepwise decrease compared to density gradient and other methods. So that's really interesting, but we can go a step further and think about selecting the single individual sperm prior to ICSI. And if we know that that is DNA intact, that should improve outcomes. Now, some of you may be using IMSI. You may have used IMSI in the past and abandoned it. There were some reviews that suggested that the clinical use of IMSI wasn't really well supported by the data. But there's some really interesting information out there. This paper from Berkowitz and colleagues it's quite old now, but what it demonstrated was that it wasn't so much about the number and the size of the vacuoles, which is what we looked at generally with IMSI when it was first introduced. But what they demonstrated was that it's actually the location, particularly the equatorial segment, and the depth of the vacuoles, whether they are centrally in the head of the sperm rather than peripheral, that makes a difference. And actually they use this system that they call copy number variation that linked to the likelihood of that sperm being poor quality, carrying DNA damage and giving poor outcomes. So there might be some scope for re-evaluating IMSI, and the most recent review, and it's not particularly recent, but it's the most recent review that I'm aware of, suggested that actually IMSI might help sperm selection and may improve late paternal effects. And we know the sperm impact late paternal effects much more than we did a while back. Now, you'll know we have an interest in hyaluronan binding. We know hyaluronan binding is linked to, amongst other things, the risk of DNA fragmentation in the sperm nucleus. This is based on the work that people like Gabor Hussar and Denis Akas and others have done, principally Gabor Hussar. And we can use this association between hyaluronan binding and DNA fragmentation to deselect sperm. You know where I'm going with this with the PICSI. So this is I use this slide all the time. I absolutely love it. It's a great little demonstration of how hyaluronan binding deselects DNA damaged sperm. So this is from Yagchi and colleagues. It's 15 years ago now, but they labeled DNA intact sperm with a green fluorescent dye, and the ones with DNA fragmentation fluoresce red. And what you can see on this hyaluronan plaque is that 99.1% of them that are bound inside that plaque are DNA intact, whereas outside it's actually 55%. So you get a much better chance of picking DNA intact sperm this way. And that's the principle of the PICSI dish. So in this multi-center study from Morroll and colleagues, what they demonstrated here was that there was a reduction in the risk of early pregnancy loss, particularly in men with low hyaluronan binding in their native semen. When you use hyaluronan binding during the ICSI procedure to select sperm for injection, you got much lower miscarriage rates. And that was the same sort of finding that the HabSelect study in the UK found. So this was unselected population of patients. So we weren't looking for particular types of patient. It was unselected. If you were having ICSI, you could have PICSI if you were in the right arm of the trial. This was close on to 3,000 cycles. And again, there was a little increase in the live birth rate, but not significant, but there was a significant reduction in the miscarriage rate. And this study, the data from this study was followed up by Western colleagues with the group there in Birmingham. And what they did is, because the miscarriage reduction was a significant secondary outcome of the HabSelect study, what they did is they did a mechanistic analysis and deliberately enriched the study for miscarriage outcomes, and then plotted the data this way. And what you can see here is that, ignore this one for a moment, for female age, what we see is when the woman becomes pregnant, this is the predicted live birth rate once the woman is pregnant. And what you can see is with age, they are at higher risk of miscarriage. In other words, the predicted live birth rate goes down. We're all aware of that. But what the West study showed is that if you use hyaluronan selection, so PICSI, in these patients, if they became pregnant, their chances of pregnancy outcome was maintained. You did not get the age-related decrease in predicted live birth rate. So they had the same chance of carrying that pregnancy to term as younger patients. And I've already said that we know that egg quality is linked to that ability to repair some DNA fragmentation. So in younger patients, it's less of a problem. In older patients, it becomes an issue because oocyte quality is reduced, and PICSI, or Hyaluronan selection of sperm prior to injection, can offset that risk. There is an impact of male age, but that's much less significant. So we'll park that one for now. Okay, so that was a quick whiz through. We know sperm structure reflects function. We know that that's carrying half the embryonic genome, and it has to deliver that to the oocyte to get to fertilization. But importantly, the key message, I think, is that we know the sperm DNA has an impact on reproductive success way beyond fertilization. That's when I started. All of our work on sperm function was around how do we make sure we get fertilization. We've moved on. We've moved on because ICSI offsets that risk quite a lot. And what we want to know is how we select the best sperm, because we know the sperm has a significant impact on late paternal effects. And we're talking about blastocyst formation, we're talking about implantation, and we're talking about early pregnancy loss. So we can optimize sperm separation using things like Zymot and careful density gradients and good swim ups and so on. We know lab procedures can impact DNA fragmentation negatively with poor handling. And we know that selection of the right single sperm for ICSI can improve clinical outcomes. And we've mentioned IMSI, but I think the data is perhaps more robust for Hyaluronan selection. And we should be mindful that all of this ultimately is related to female age and oocyte quality, because it's the women who are older and or have compromised oocyte quality that will benefit most from us being very, very careful with the way we handle, prepare and select sperm before we use them in treatment. A quick heads up, Steve Fleming and Martin Nace, my colleagues in the professional education and clinical support team, have published a paper on sperm separation and selection techniques to mitigate sperm DNA damage. You can have a look at that, which covers a lot of what I've spoken about today. And those are my selected references, and I will leave it there. So thank you very much for listening.
Thank you, Dave, for a very informative lecture. It was very informative and a lot of information. Now we're moving to the Q&A question. And if anyone has a question, can you just write it in the QA box, and we can answer as many questions as we can. First question, Dave, what is your experience with sorting semen for a short period of time, 48 hours before processing, and using for ICSI? And does it affect on the DNA fragmentation and embryo outcomes, pregnancy? And sometimes he put the semen sample at 2 to 8 degrees in the fridge. So what do you think about this?
Yeah, it's not something I've ever done. I'm desperately trying to remember some of my earlier reading around putting sperm at lower temperatures before preparing it. Yeah, I'd have to go back and check that. I'd be cautious, and I'd be cautious because most of the data suggests that preparing your sample as quickly as possible after production and then using it soon after preparation seems generally to be preferable. But I would need to go back and look at the literature, to be honest. Yeah. Yeah,
okay. Where about the sperm freezing technique? Is it like if we have like a fresh semen sample, if we have the ability to have a fresh semen sample or frozen sample, which of the semen sample would be affect on the DNA fragmentation? Is sperm freezing a process does affect the DNA fragmentation?
It certainly has the potential to do it. And one of the things that I'm particularly mindful of is there is a really nice paper from the EV group that Marcos Masegwe published. and it was really around donor oocyte quality. That was the principal aim of the study. But what was interesting about it was what they showed was that if you look at DNA fragmentation after a swim up from a fresh sample, you get that real reduction in DNA fragmentation after preparation that I've talked about in the talk now. What they showed was that if you do the same thing on a frozen sample, the level of recovery of DNA fragmentation is not the same. So after freezing and thawing, if you do a swim up, you don't get the same reduction in DNA fragmentation that you do in a fresh sample. So I would keep that in mind. That doesn't mean there aren't other ways of doing it, but I'm just wary that DNA fragmentation after freezing and thawing may not behave the same way.
Okay. Another question, you're talking about the MC. Has the sperm head vacuole been proven to correlate with DNA fragmentation and improper DNA packaging?
I think there's a degree of assumption that they are indicative of some problem with the way the sperm head forms. Now, again, I'm racking my brains and I can't think of a paper that directly shows that the vacuole is linked to a portion of DNA that is damaged in some way. But I think it's more of an association between two things: a physical manifestation of a problem in the sperm head, which is the vacuoles, and the bit you can't see, which is the DNA fragmentation. So I can't think of a paper that explicitly shows that the two are the same thing, but there is certainly an association.
Okay. Other question, there studies that show significant improvement in sperm DNA fragmentation after lifestyle and diet changes and clinical outcomes? You know that the physician recommends some diets and capsules, and what do you think about this?
Yeah, really interesting, and I think we're all aware that one of the big issues with any study that looks at improvements in sperm quality is fraught with problems with how you count them, how you assess motility. The variability between DNA fragmentation tests is a similar issue because of the standardization of tests. So it's always quite difficult to take out the methodological issues. There are some good studies showing in men with persistent levels of high DNA damage that oral antioxidants can be helpful. And I think there's also a reasonable body of evidence to show that if you've got a good diet and good lifestyle, so you're physically active, you eat well, then it's not only good for your general health, but your sperm quality can improve. And the corollary of that is that, of course, if you don't eat well and you eat too much, we also know there's a strong association between obesity and outcomes for men reproductively, but also interestingly, that is passed on to the offspring. So the boys born to men who are overweight and obese, they also have issues potentially. Some really nice data presented, I think it was ESHRE last year. So yes, I think the quick answer is yes. There's a good body of evidence out there that you can improve outcomes if you take antioxidants, but the antioxidants are really an alternative to just a good diet and lifestyle.
Okay, many questions popped up on the Q&A box. One of the questions is, it is clear that thymus increase the euploidy rate, but does it help in translocation from man side? the translocation that happens in the chromosomes?
Yeah, this is where I'm teetering on the edge of my genetics knowledge. The data that we're aware of for the zymote is purely the broad sweep of aneuploidies reported in PGTA cycles. So translocations, again, I'm racking my brains, but I can't think of any specific papers that deal with that.
Okay. Other question, if we have like TC sample, would you select perfect morphology, very poor motile sperm over a poor morphology with a good motility?
That's in a TZ sample.
Yeah.
Yeah, that's interesting. Honestly, I don't know. I don't know scientifically what is out there that can say categorically one way or the other. I'll give you an opinion. My opinion is that the motility is useful primarily as a way of determining that the sperm's alive. Morphology is useful up to a point, because what it will show you is how mature the sperm is based on the amount of cytoplasm that's still attached potentially, but also the shape and size of the head. But what you're really doing is just making sure it's alive and the head size is about right for a mature sperm, and that's about as much as you can do, in my opinion. Bearing in mind that we know TZ might be slightly different, but we know in the men with severe oligoasthenoteratozoospermia that even their morphologically normal sperm that we tend to pick out for ICSI, they have elevated risks of aneuploidy and DNA fragmentation.
Okay.
So it's a guide, it's not diagnostic.
Okay, there's a participant asked for the link of the recent paper again for Martine, and so we can provide her with the link or the paper she asked for the paper.
Absolutely, yeah.
Another question, many questions coming, Dave. Where about the second semen sample on OPU day? Instead of having abstinence, having a second semen sample.
Yeah, again, I know some labs have tried that, and I've tried it myself when I was linked to a clinic between Liverpool and Manchester in the UK. And we, in the end, we abandoned it because we couldn't see any clear difference. It's a little bit more, I know the men get an easy part of an IVF cycle, but for the man, it's a little bit more stressful to produce 2 samples on the day. And I am not convinced that it's any more useful than having 1 or 2 days abstinence. But again, that's an opinion. I'd have to trawl back through the literature, but I'm not aware of anything that shows that two samples on the same day will actually improve above 1 day's abstinence, for example.
Okay, another thing that you mentioned before, is it better to do everything at room temperature and not use an incubator for sperm? Also, should you bring the sample to 37 immediately before ICSI?
Again, I'll give you my opinion. So just take that however way you want to. I would do everything in the sperm lab at room temperature. So unless semen will liquefy at room temperature, usually. So you could have an incubator or a warming oven available for the occasional sample that liquefies poorly. You might put that in an incubator for 20 minutes or something. But for the majority of samples, you can just do everything on the bench, and I wouldn't put anything at 37 degrees. And then, so I would keep it at room temperature. I would do the gradients or whatever it is at room temperature. Just be aware that at the moment, at least, the instructions for use for things like the Zymote say incubate at 37 degrees. You would typically do a swim up at 37 degrees. That's a whole different discussion, which I'm happy to take if necessary. And then I would only warm up a sample if it was conventional IVF, where I would put it into a fertilization medium and I would pre-warm it, pre-gas it for 30 minutes before I use it. For an ICSI, I wouldn't. I would just go from room temperature into my PVP drop.
Okay. Where about the PICSI? Could PICSI help in minimizing the sperm DNA fragmentation? Talking about the PICSI, do you have any, like, how do we optimize, Dave, in your opinion, doing zymote and doing the PICSI at the same time? When we're going to use zymote, when we're going to use PICSI?
They are two complementary things. So they could be used together, or you can use one or the other. The PICSI you can use after any sperm preparation method. And what the PICSI is doing is it's allowing you to choose a sperm that you are actually injecting into the oocyte at minimal risk of it carrying DNA fragmentation. We can't say it's zero, but from that Yagchi paper, for example, we know that it's over 99% chance that you're going to pick a DNA intact sperm. What the zymote does is it will bring the DNA fragmentation levels in your post-preparation sample way, way down. And depending on the paper and how they measure DNA fragmentation, it looks like a stepwise decrease compared to a standard swim up or a density gradient. But you are still selecting a population of sperm that you can then use in treatment. So there's still a risk, even if your post-preparation DNA fragmentation goes from 20% down to 2%, you've still got a 2% chance of picking a DNA damaged sperm if you do nothing else. What the PICSI will do is it will take that 2% chance and take it down to 0.2 maybe. So it's a stepwise decrease. So they are complementary, or you can choose to use one or the other.
Okay. Where about they talking about the zymote? Is it advisable to prolong and extend the incubation for more than 30 minutes to 1 hour or 2 hours in zymote to retrieve more motile sperm?
No, there's no evidence of that at all. The early work that was done on the use of this porous membrane strongly suggests that if you get to 30 minutes incubation, you really plateau in terms of its selective efficiency. So you've got to balance the drive to increase numbers against the selective nature of the membrane. And if you go longer, you might get more sperm, but your selection decreases because it's the dynamics of the best sperm get through first. So if you extend the time period, first of all, it didn't look like you get many more sperm coming through, but if you do, it's the poorer sperm. So we generally say don't go beyond half an hour. There's no great merit to it. If you need bigger numbers, so for example, if you feel you're not getting enough, you should get enough with a decent sample. But if you've got a borderline sample or you want bigger numbers to do IUI with, then you can always opt into using a density gradient instead. But I would urge against going beyond half an hour for the incubation on the zymote.
Okay. There's which method of sperm DNA fragmentation test you recommend?
I don't. I'm not saying don't test for it. I just think there are pros and cons of each one. You know, you can buy kits that will allow you to do the halo test. So if that's the only one available to you, then you can use that. There are others like the sperm chromatin, the ICSI, where there's more data and it looks more sensitive and more specific, but it's much more expensive to do. So as long as you use the right test for you and interpret it in the right way, I wouldn't be inclined to say definitely use that one rather than that one. But you've got to be wary of what the different tests are telling you and how you use that data.
Okay, I have like final two questions. The first question, impact of varicocele on sperm DNA fragmentation.
Not really an expert on varicoceles, if I'm being honest. I might be better answering that by email.
Okay.
But my gut feeling, my response from the bit I know, is that varicoceles vary considerably in their impact on fertility generally and sperm quality. So there isn't an absolute answer to that, I suspect. But if we know who submitted that, I'm quite happy to try and answer it by email.
Okay. We have a lot of questions, and we're going to answer it later if we don't have time. The last question, Dave, would you prefer max over zymote or what?
Would
max or zymote?
Max.
Oh, yeah. Magnetic.
See, I could get myself into trouble here. I've visited a number of labs that use max, and I am not convinced it is worth the time and effort. I would rather do. I would rather use a zymote, well, let's answer this a different way. I showed you the paper from IVF Brussels by Matezol and colleagues from Nilke de Monk's group at the lab there. They have used max. They have used a number of different methods, and they have adopted the zymote for their routine sperm preparation. So that, to my mind, answers that much more clearly than me giving an opinion. Okay.
Well, we'll end of this segment, the Q&A segment. Thank you so much, Dave, for wonderful answers. And if you have any inquiries about this presentation, you can send it to us and we can provide with the answer. Thank you for joining us again, Dave, and for the participants also. And stay tuned, please, for our next webinar. It's going to be on June 19th. Thank you so much, Dave, for your time. And I'm hopefully that they will be happy with the presentation. Thank you.
Thank you. Thank you for
so
listening in.
Thank you.